Bio 133's Test 1 review. I'm going to start off with some study strategies for Test 1, and then I will proceed on to kind of go over some of the important features of Test 1's chapters, Test 1, 2, and 3. Now, you're going to see a different screen here in a second in terms of some study tactics, and you'll see me write on some things, so that'll help you also, and you obviously can hear my voice. So I'm going to go to first. Just go back and back here so you can see. This is what you would see if you opened up Mastering Biology in terms of the page that we use page that we use for our assignments. So if you go to over here and look, you'll see study area, and you click on study area there, and you click on access study area. You then can have some different study tactics other than just reading and reviewing your notes, etc. Each chapter you can click on. You can see I can go into chapter 3 here like this, and my ebook is there, as well as some videos to watch of some stuff that we've talked about from chapter 3, as well as some practice quizzes, which are great to see if you know what's going on in terms of that particular topic. And this answer key also shows you the answers to the back, or to the answers at the end of each chapter, which is a really good idea to do as you're studying for the test. So just an area to go to other than what you would see, from your textbook and your notes and stuff in terms of that. Okay, so I wanted to show you that. I'm going to show you a couple of videos too, in addition to, oops. Nope. Find where. There we are. In addition to, let's make sure I'm still broadcasting here, what I'm going to go through with each chapter. Now, one of the things I like to do when you talk about each chapter is just kind of look at the big picture things that you would be thinking about with each chapter. Now, first of all, one of the things that you want to be able to do when you think about, let me get rid of this, you don't need to see this. When you think about studying for a test, is you want to think about, we've obviously had 3 chapters, chapter 1, chapter 2, and chapter 3. And if you look at the review sheet, you see the objectives from the review sheet that you should be familiar with. Now, each chapter, obviously, you want to think about, what did we spend the most time on? And that's where I'm going to, obviously, as an instructor, kind of assess you in terms of question-wise. Well, I'll spend the most time on writing questions. And so as you look at your review sheet, you certainly want to be able to think about all the things that you see on the review sheet, but then you want to really narrow it down and go, okay, for chapter 1, if I were to summarize my notes, or for chapter 2, or for chapter 3, what main things that we spent the most time on, and you're thinking also about, did we do worksheets? Those are things that if we did a worksheet, whether I had you hand it in or whether we went through it together, which pretty much we did in class, you're using those to study, because obviously I took time to make you take the time to think about that. So those are good pieces of the puzzle as you're thinking about, test questions and what I might ask in terms of that. As we talked about in class, there'll probably be between 60 and 65 multiple choice or true false questions that obviously are divided amongst the 3 chapters. Now, in reality, obviously, we spent a lot more time on chapters 2 and 3 than we did with chapter 1, but there will be still a few questions on chapter 1. So that's just kind of an idea, that you can think about in terms of, as you're strategizing in terms of the questions that you would see. Now, for chapter 1, oops. There were probably 3 main themes that we spent the most time on: the characteristics of life, the defining features of humans. We briefly also talked about the levels of biological organization, smallest to more complex. And then we looked at the scientific method. So, real briefly, you'll hear me go through this kind of quickly because this is a review session and I want to get this out to you so that you have it. It's not going through it just like we did in class, but, in a little bit more quicker pace in terms of that. So for the signs of life that allow us to know that we're alive, these 7 things would have to occur in terms of that. And so if you looked at a rock versus, an animal or a human like us, you would see these 7 things that we're composed of molecules. And we talked about, the biomolecules or the organic macromolecules that we identified in chapter 2, carbs, lipids, proteins, and nucleic acids. Now, in and on their own, they're obviously not alive, but when we start putting them into pieces and parts of cells, and we have to utilize those in a living body, we know that we're alive based on that. Now, requiring energy and raw materials, obviously energy, we need to use some of these biomolecules to help us make ATP for our cells. We obviously have cells, which are the basic unit of life, and there's different types of cells in our body as we started to investigate that in chapter 3. Let me erase this. Basic unit of life. Maintain homeostasis. Remember we said you want to be able to maintain a stable environment like temperature, blood pH, blood glucose, heart rate, blood pressure. I'm going to abbreviate environment ENV. Those are types of things that we would internal environment that we would think about when we were maintaining homeostasis, and a living individual has to do that. Responding to the external environment, like I gave you an example of when your pupils that let light into and out of your eyes to get to your retina, your pupils can constrict when there's too much light, like if you're squinting in the sun, and they can dilate, get big if you go into a darker room to let more light in. So they're responding to the external environment. We're able to grow and reproduce, and we're capable of evolving. So those are the things that we talked about in terms of being alive. And then the next feature we said, there's 4 features that allow us to think about the fact that we are humans. And of the 4 features, most of us here would understand that being bipedal means we can stand on 2 feet, and that frees the hands for being able to grab things, which then allows us to have had more technology and become a more complex society in terms of that. So standing on 2 feet, the advantage is freeing the hands. Opposable thumbs, if you take your hand right now and you take your thumb, you can actually touch all 4 fingers or digits. And your thumb, to be able to do that, opposable thumbs, is important because it allows us to grasp things more easier. And again, that can help if you think about how we've evolved as a society over hundreds of thousands of years to be able to build things and et cetera. Large brain, to not only be able to have the ability to speak, but making complex decisions. So this obviously allows us to be, thought of as humans. The other big piece to chapter 1 was the 5 steps of the scientific method. If you recall in class, I gave you a newspaper article regarding the Buffalo News and a pretty rare example of a woman that had auto-brewery syndrome, in which when she ate carbohydrates, her digestive tract had possessed a lot of a microbe called yeast, and allowed her to, unfortunately, turn the normal carbohydrates into ethyl alcohol, which allowed her to become intoxicated, even though she wasn't drinking alcohol. And through a process of going through these steps of the scientific method, we talked about things just like that we could make sure that, that was a true thing. Most people probably didn't believe it. So in her case, as we talked about, we observed and generalized the fact that she got pulled over for a DWI, but she basically said she did not have enough to drink to have a DWI. And so at first, she obviously got a lawyer, and her lawyer observed and generalized, like, you know what, there's no way you should have been drunk. Let's formulate a hypothesis. And the hypothesis in her case, and the educated guess, was perhaps there was something else causing her to become intoxicated. So we had to make a testable prediction. And after looking through some literature, her lawyers obviously found something called autobrewery syndrome, which is very rare. But again, this is why you go through the scientific method. And, obviously this is a very rare case. We talked in class about, blood pressure and developing new drugs, but I think this can kind of make you think about it too. And then they designed an experiment in which she had to go into 2 different testing centers, be given carbohydrates from, somebody else that wasn't her lawyer, take blood levels of alcohol before and after, and lo and behold, they found out after they did the experiment that their hypothesis was correct. So they didn't have to go back through the scientific method again. Now, certainly if they were wrong, they would go through the steps of the scientific method again to go ahead and figure that out in terms of that. Now, those were the 3 major pieces. Now, obviously, along the way, we looked at examples of another, here's a study of blood pressure. We talked about experimental groups, control groups, what blind and double blind meant. So you can go back to your notes. Again, this is just a way that, you can look at to see the big pieces that we actually talked about in chapter 1 in terms of that. We also briefly talked about this Dietary Supplement Health and Education Act. While you wouldn't have to remember all these different popular herbal remedies, food supplements, and hormones, I kind of just gave you this as an eye-opener for you to become a skeptical thinker. So there's chapter 1 kind of, big picture things that you would want to kind of look at. Now let's look at chapter 2. And chapter 2, we kind of broke up chapter 2 in half. Remember, we had a worksheet that kind of looked at, if you recall, our goal. Oops. Let's go to their sketch pad here. Erase letter A. Was to think about, the atom, the element, the molecule. This was the non-living part of things. This was the beginning of chapter 2, and we did a worksheet on this just to learn the basic features and pieces and parts. And then we talked about how molecules can be put together in a specific way to form the living basic unit of life. And now, when you have common cells put together, you have tissues, you have common tissues put together, you have organs, and common organs put together, you have organ systems. And this would be, where we're headed to for the rest of the semester. And these would be obviously the living room based on that. So we kind of went through this type of idea. So the beginning of the chapter covered this. And, we talked about the fact that you wouldn't have to know this periodic table, but then some of the more common, elements, the atoms make up the elements that you see here. And like, for example, oxygen, hydrogen, calcium, sodium, potassium. These are some more important elements that eventually can be put together in molecules. These elements really can't be broken down into a simpler form, but there's atoms that make up these elements. And so then you have the elements when you put them together. We gave examples such as, if you put, carbon, 6 atoms of carbon, 12 atoms of hydrogen, and 6 atoms of oxygen together, you got glucose. And so you can see the 3 elements, and glucose is a molecule, and we need glucose as we'll mention briefly in chapter 3 to eventually help us make ATP. So this is an important feature. And then we also learned, obviously, that there were 3 different parts to an atom. There were in the nucleus, there were the protons and neutrons. The protons were positively charged, the neutrons were negatively charged. That made up the atomic mass of the atom. And then you had these electrons that floated around the outside, which really are what's going to determine our electrical behavior of atoms, which remember, we referred to that as being important in terms of forming chemical bonds. And recall, I won't write them down, but you can recall, we'll see them shortly here, the 3 types of chemical bonds. So you could put these these atoms of these elements together in a stable molecule. You had the hydrogen bond, you had the ionic bond, you had the covalent bond, and we'll look very quickly at those again. But what was important about these electrons was especially the ones in the outermost shell or the valence shell. Those are the ones that can interact with atoms of other elements and then start to form some chemical bonds that you'll see in terms of that. And that's kind of why we really needed to sort of look at these atoms and think about that, okay? So the electrons are the ones that are important. And remember, there were 3 types of bonds, right? And remember, of the 3 types of bonds, the covalent bonds were the strongest, the ionic bonds were moderate, and the hydrogen bonds were the weakest. So when you look at the covalent bonds, remember I said there's one word you want to think about with these valence electrons, you know, because remember, it's the electrons that are going to help me form these chemical bonds. And the one thing that you're thinking about with covalent bonds is in those valence shells, those atoms of those elements are going to be sharing electrons. Now, I talked about in class that you didn't have to give me examples of when covalent bonds share electrons equally or when they share electrons unequally, but obviously two common molecules that we've discussed a little bit in class, water and glucose. You can see, for example, water, it's 2 hydrogens and its oxygen atom, which you can see here. The outermost valence shells are sharing the electrons to try to complete their shell. When they complete it, then that forms a chemical bond. We're not going to get into specifics of what numbers of electrons need to complete an outermost shell, but there's a certain number in some of these elements that you have to have a certain number to complete that and make the atom or the element stable, which in reality allows this to stay together. Same thing with the carbons and the hydrogens and the oxygens. And typically we're seeing examples of what we call polar covalent bonds, which means the electrons aren't really shared equally, but there's still a sharing to hold them together, these atoms of these elements, so that we form molecules, like you can see water and glucose here. Now that's the strongest covalent bond. The next size covalent bond we had was the ionic bond, and this is where we talked about, you know, electrolytes in our body, like sodium and potassium, and these can also be called ions. Well, they have a charge, and so instead of sharing electrons, for example, that sodium atom and that chlorine atom, notice the arrow there, that electron is going to be transferred to the chlorine atom. Now notice sodium now in its outermost shell doesn't have any electrons, and so if you counted the number of electrons, remember electrons are negatively charged, but inside the nucleus there, you still have positively charged protons. And so now there's more positively charged protons than there are of the rest of the electrons that are surrounding the sodium atom. And there's more positive charges than negative charges, we get a positive charge. When the negative electron gets transferred to the chlorine atom, we have 1 extra electron than we do, more negative charges than we do positive charges. And that gives us the negative charge, and these two attract. Opposites attract, and to form our common, what we call table salt. Now, if we stick table salt in a watery substance or solvent, something that can dissolve it, we get ions, and the ions pull apart. And in our body, we have water inside our cells, we have water outside our cells, we have water in our bloodstream, and these ionic bonds can break apart, and then we have our electrolytes that do have specific functions in our body. So you can see the ionic bonds here, the second type of chemical bond. The third type of chemical bond exists in water. Remember, it's our weakest bond, and we talked about this guy in class. It also exists between DNA and proteins. So these were our 3 chemical bonds very quickly. We then talked about how important water was, and we said we were almost 60% by weight water, and most of that water was in our cells, which you're going to hear me talk a little bit about when we wrap up some stuff from chapter 3 very quickly. Again, we're moving along quick because this is a review. If my computer will let me write on this, which sometimes it's fussy. There it goes. So even though what it's saying, but every cell is 75% water, it's basically saying 60% by weight is water, and most of that water is found inside a cell. So if cellular volume changes, we talked about dehydration and water intoxication, problems can result. But water dissolves things. biological solvent. Water is a lubricant. Water helps us maintain temperature, and obviously then water is a big chunk of our cells in terms of that. Now, we also spent some time with pH, and we made some clinical connections with pH. Again, I'm not going to go through everything, but just as a quick review of your pH scale, remember the lower the number, the more acidic, more hydrogen ions you had, and the higher the number, the less acidic or more alkaline or more basic you were. So more acidic, more basic. This meant as the number goes down, you have more hydrogen ions. That's what the H is. And as the number goes up, you become more basic or alkaline, and that means there's less hydrogen ions. It's less. We basically say it's less acidic. And we talked about the different, types of pH values that you'd see. Most of our human, obviously the human bloodstream and tears and most of the places in our body that have fluids typically are around the neutral range, where there's equal numbers of hydrogen ions and a special, remember we taught a base ion called OH. And we talked about the fact that we wouldn't want our blood to get too acidic, and we wouldn't want to get our blood to get too alkaline, and our body does things to help us stay at homeostasis in terms of that. So that was important. And there was a little bit on the worksheet in regards to that too, that you were able to answer. Again, look at your worksheet. Then the second half of the chapter involved, remember we did a concept map on the 4 types of biomolecules. Remember, we also called these biomolecules, and refer back to your notes for the concept map. Again, I'm not going to go through each specific thing like we did in class because we already did that. But when you're thinking about the biomolecules, remember we spent a lot of time on carbohydrates. There were 3 different types of groups of carbohydrates: monosaccharides, disaccharides, and polysaccharides. And then you were able to give specific examples. Like glucose is a monosaccharide, whereas starch is a polysaccharide. And we talked about, what kinds of functions you'd have for that. Lipids, the same thing. There were several different categories. What were the functions? Proteins, think about the building blocks of proteins, the amino acids, and what the different functions are for those. We said we really didn't, consume nucleic acids as a food. And obviously we do carbohydrates, lipids, and proteins. These macromolecules or biomolecules also exist in our body in terms of how we utilize them. But remember, our nucleic acids were our DNA and our RNA. So that was a quick run through of chapter 2 and some of the important features that you would want to look at. Again, this is just a strict overview because we did some of these things obviously in class. And then finally, chapter 3, and I'm actually going to show you a couple of videos in regards to this too, to help. This was the most recently covered chapter. Remember we initially talked about how there are different types of cells, and even though usually cells are, drawn like a circle, you have really, in reality, cells that look different than just circles. You have, heart muscle cells that are branched that can contract. You have nerve cells that look like microfibers. They have to generate electricity. And then you have like kidney cells that are like little cubes in order to process and make urine. So even though a cell can look different, it's still going to have the parts about the organelles that we worked on and talked about using our worksheet. But its function might be different in terms of what it's supposed to do. Muscle cells in the heart are supposed to contract. Nerve cells are supposed to generate electricity. And kidney cells are supposed to help us produce urine. Remember, we started off this cellular kind of visit talking about the cell membrane. You'll probably see that overviewed in the video here shortly. And we didn't talk about transport right away because we first wanted to talk about what was inside of a cell. Again, this is where you'd want to refer to your worksheet. There's 2 things that you want to think about when you are referring to your worksheet. You want to first think about, could I label figure 3.14? Now, I'm not going to ask you to label it, but there could be a multiple choice question. Like, for example, if I were to point to that structure, maybe this is a question 46, I'd say, what is that question? What is question number 46 pointing to? And you'd say, okay, there'd be A, B, C, and D for your choices. And obviously one of the choices would be a mitochondria. So you'd hopefully choose that one, in terms of that. The second thing you want to be able to do is understand, identify the organelles as well as know what their functions are, like we did with our worksheet. And that's going to be important. I'm going to show you a quick video so that you're able to see that. And I think it'll help. And then we went through and did a little activity in class in terms of trying to differentiate between passive and active transport, as well as what bulk transport is. And I have a little video kind of review of that also that I'm going to show you. And we talked about the differences in terms of, do I need energy or don't? Remember, we categorized the transport processes. And, look at your notes from class. We asked ourselves, if it was passive or active transport. We asked ourselves 3 questions. Do I need energy? How's the concentration gradient? Which really means how are things moving? Okay. And that says concentration gradient. That means, are we moving things from high to low, or are we moving things from low to high? Downhill is high to low, uphill is low to high. And then the third question was there different examples or types? And then we went through these today. So again, look in your notes. You're going to see my video, in terms of that. And we did mention also, about bulk transport, that it's a third way to move things across cell membranes. It's not necessarily passive transport, it's not necessarily active transport, but when we need to move big things either into the cell or out of the cell, I gave you an example of like Wegmans, when the big trucks going behind and not going through the front door, we use bulk transport. So it's a third type of transport that you would see in terms of that. Now remember, give me 1 second, I just have to check on something here. Give me 1 second, excuse me. Sorry about that. So remember in class we talked about that there were 3 different types of passive transport. Remember, transport in which you don't require energy, which we're moving things from a higher to lower concentration gradient, across the cell membrane. And one of the ones that we talked about was osmosis, which is just water. So the special movement of water, no energy required, from a high concentration to a low concentration, either into a cell or out of a cell. And we need a special protein channel to do that because water is water soluble and it has a hard time getting through this lipid bilayer. Well, osmosis has some ramifications in terms of what can happen to a cell. Remember, most of our body water is found inside our cells. And so we didn't get to talk about this in class regarding chapter 3, so I'm going to mention it now. There's a word called tonicity, which really means cell behavior. And by cell behavior, do we mean, does the cell shrink, get smaller? Does the cell swell, get bigger? Or does the cell stay the same size? Now, in reality, we do not want this to happen to our cells, what I circled, to shrink or swell, because that could compromise the organelles inside and the cells, function. So we start to think about cell volume or cell behavior, tonicity. And when we do, we see these terms, isotonic, hypertonic, and hypotonic. Notice the tonic suffix, or the ending of the word, referring to how does the cell behave. Now, in our bodies, an example using red blood cells, if this is a normal red blood cell, which you can see right here, which can carry oxygen and carbon dioxide for us, we talked a little bit about that. The red blood cell itself, it sits in our bloodstream, okay? There is fluid around the cell. We call that fluid extracellular fluid, right? And now that can change. Inside the cell, this is where we could call this intracellular fluid, ICF, or we could also call it cytoplasm or cytosol. We learned that this week. And so what is this? What is this tonicity thing? Why should we be worried about it? Well, me erase this so you can see. When you think about the fact that this extracellular fluid can change around the outside of a cell, you start to think about a couple clinical conditions. You start to think about, what if a person got dehydrated? I'll abbreviate. Or what if a person drank too much water, which we briefly talked about a few weeks back, water intoxication. Can that compromise the amount of fluid inside a cell, like a red blood cell? Well, so if you look at my pictures over here, you'll notice right away that this isotonic means, iso means same, tonic means cell volume. So notice my arrows here. And what it's alluding to is, this is how we want, what we want cells to be bathed in. We want cells to be bathed in an isotonic solution. We want our extracellular fluid to be isotonic, because if it is, the amount of I know this isn't in a cup of fluid, but imagine the outside of the cell, the extracellular fluid, okay, is isotonic, meaning it has the same amount of water and special particles called solutes, like ions or proteins, etc., as what's inside the cell. It's almost exactly the same, or it is exactly the same. And that's what we want our cells to be bathed in. We want that. So that as you think about osmosis, water is always moving from a high concentration to a low concentration. But why don't our cells normally swell or shrink? Because our cells, the outside of our cells, the extracellular fluid of our cells, are bathed in a solution that's isotonic, which has about the same amount of water and special solutes or electrolytes as what's inside the cell. So the ECF and the ICF, or the cytosol, are equal. That creates a situation in which osmosis is occurring, but there are equal amounts of water moving in and out of our cells, and our cells stay the same size. And this is what we want to happen, okay? We don't want the other two things to happen. Stuff that you put into your body. For example, eye drops. If you are a contact lens wearer, if you like to put eye drops into your eyes, if you look at the eye drop bottle, you don't want your eyeball cells to swell or shrink. So you put something that's isotonic into your body, so that doesn't change your extracellular fluid and keeps your cells relatively the same size. This is osmosis at work. This is what normally should happen. Equal amounts of water moving into or out of a cell from a high concentration to a low concentration. No one knows the better that this is happening, but our cells don't swell or shrink. They stay the same size and we're good to go. But now, what if we flip the tables? And I'm going to erase some of this so that we can see. Remember my scenarios. What if my extracellular fluid changes and I become dehydrated, and that causes a change, or I get drinking too much causing water intoxication? Okay. Now, a simple test with red blood cells can show this. Again, we don't want this to happen, but let's look at this example right here. Notice my red blood cell, when I stuck it into something that was hypertonic, is shrinking. It's shrinking. Well, how the heck did this happen? It happened because the extracellular fluid around that cell changed. We lost water. Maybe we were sick for several days, stomach flu, vomiting, diarrhea, and we lost water out of our bodies. Maybe we sweat profusely because it was really hot and we lost water out of our bodies via the bloodstream. And when that happens, it creates a situation where there's less water around cells now, and the extracellular fluid that's surrounding these cells has become compromised. And so with that case scenario, notice my arrows here. There's more water moving out of the cells here than water moving in. There's a real small arrow you can see right here. More water moving out of the cells, water osmosis, high to low concentration, than water moving in. Guess what? It's unequal water movement. We don't want osmosis to do this, but it does it randomly. And when that happens, guess what happens when you get dehydrated? Your cells will shrink. Not good. Typically, when your cells shrink, you get symptoms. Your mouth gets dry, you get a headache, you feel thirsty, you hope to drink some fluids to replace what you've lost. But what if it's a serious situation? That's when you need to go into the hospital and get fluids. And guess what kind of fluids you get? Isotonic fluids in a bag. You can even see that on your bag, the little bag that they put from a tube to a needle into your arm. You see that? It's isotonic, so it won't affect the size of your cells, and our cells won't continue to shrink. And dehydration can hurt you, can kill you, in terms of that. So you want to be careful with that. Now, what about the opposite part of this? Let's erase this here. Give me one more second. I'm in my house doing this right now, and most of you know that own animals. I have a dog right now that's barking its head off, and I'm not sure why. So just excuse me for about 10 seconds, station break here, and I will be right back so I can see what mischief she's in. Okay, sorry, all is well here. So I don't know what the heck was going on out there, but just checking. All right, so what if, now we're talking about the what if we drank too much water? Again, remember, we started this off saying we want our cells to be isotonic in terms of what surrounds the extracellular fluid. We don't want either of these two situations to occur. So now we're looking at this guy. Whoops. Okay. Now, what happens if our cells have a situation where they become hypotonic around them, the extracellular fluid? What really means is there's too much water that surrounds the cells. And I would sometimes have to purposely do that by purposely drinking too much water too quickly, water intoxication. And when I do that, I create a situation around the cells where now there's too much water. And the cell osmosis tries to take in some of that water, but unfortunately more water moves into the cell then moves out. You can see the smaller arrow moving out here. And again, it's unequal osmosis when we want it to be equal, but it's unequal. And because of that, the cell will swell. You can see my red blood cell swelling there. And our normal cells can do this also. And, we talked about the young lady in California several years ago that drank too much water too quickly. And she was trying to win a Nintendo Wii for her family, and it was a stupid contest. And exactly how much water she drank. She was a thinner woman, but basically she drank it quick enough and the contest did not allow her to pee or vomit, or she was out of the contest. And long story short, unfortunately, she put so much water around her cells, her brain cells and her cells swelled, and she started to have symptoms of water intoxication, a headache and nausea and wanted to vomit. But instead of going to an emergency room, she went home and laid down, and she obviously lost the contest. And so they sent her home. And when she did, her brain cells swelled enough that she went into a coma and died. So this water intoxication can be serious also in terms of this. Now, treating dehydration, we give you water or you drink water to try to get your cells to go from shrunk to normal size. Now, if you're in a serious water intoxication situation and your kidneys have failed, which that's typically what happened to her, she just overwhelmed her organs with water, we're going to need other drugs and methods to help get rid of some of that water from our body. Maybe using some anti-inflammatories or catheterizing a person's bladder if they're unconscious to try to remove and hope the kidneys get going again to remove water. We might even have to use dialysis. It's serious. So we don't want either of these two situations to happen. And our goal in the body in terms of osmosis is to make sure that the body keeps the outside of the cell isotonic, the same amount of water and solids as inside the cell. And then typically we don't have to worry. But clinically, obviously, you can see that these things can happen in terms of what you'd see. All right. So I'm going to finish my last little bit about, we didn't get a chance to cover this, but I want to, tell you right away. When we're thinking about the last part of the chapter, making the fuel called ATP, which remember we said the mitochondria are organelles that help us produce the energy in the form of ATP that our cells can use. For example, why would we want to use ATP? We can move things across cell membranes. Remember active transport? We needed ATP for that. Okay. Well, muscles can contract. I'm going to abbreviate cell membranes. Contraction of muscles. Right? We're going to need ATP for that. Well, if I can spell right. It's hard to write on the screen here sometimes. Muscles. We talked a little bit about, electrolyte balance and active transport and ions moving and keeping ions certain amount inside and outside the cell. We need active transport to do that. And even to build things like you want to make a new molecule or make a new hormone, synthesizing things, we need ATP to do that sometimes. Synthesize. I'm going to fit this on my screen here. So these are just some examples, moving things across cell membranes via active transport, contracting muscles, building or synthesizing, like, for example, hormones. We need ATP to do this. And so remember we said one of the main forms of the biomolecules, and we break down into our carbohydrates into glucose, the single monosaccharide. The glucose is going to get into our cells and through a pretty elaborate process. This is called cellular metabolism. Okay, and that's basically taking glucose, C6H12O6, and completely breaking those chemical bonds, those covalent bonds apart. And as we break them apart, energy gets released, and that energy is going to be kind of transformed into energy that the cells can use. We can't really use glucose for energy at the cellular level. We are transforming it into energy called ATP, and we need the help of our mitochondria to do that. Now, there are 4 stages. So the idea that you're thinking about is, Professor Finn, what do we need to know about cellular metabolism? And here you go. And because your book goes quite a bit into this process, and you're not going to need to know the intricate details. So here's what you do need to know. What are the stage names? And here they are, in which glucose is going to get into a cell and go through these special stages and eventually get broken apart so that we produce ATP. The first stage is called glycolysis. The second stage is called the preparatory step or linking step. The third stage is called the citric acid cycle. It's also called the Krebs cycle, too. And the fourth stage is called the electron transport chain, or ETS, which is abbreviated. Electron transport system or electron transport chain, should see. All right, so you should know the stage names. You kind of know what this overall we're getting glucose into our cell, and glucose is kind of going through this factory kind of step-by-step factory conveyor belt, and it's got to go through these stages. And what we're doing is trying to break it apart into its most tiniest components and extract energy out of it to produce ATP. So you should know the stage names. You should know where the stages occur. So where do the stages occur? Well, it's real easy to see here. That orange thing right there is a mitochondria. So 3 of the stages occur in the mitochondria. Let me get rid of some of these things so you can see this. Right? Let me use a different color. Bruise pink. The preparatory step, the citric acid Krebs cycle, and the electron transport chain, all of these stages occur within the mitochondria. That's why we call the mitochondria our powerhouse of the cell. That was the function we learned about with the mitochondria. And we call it that because most of the ATP that we make with one molecule of glucose gets made in these mitochondria. The other stage, glycolysis, which is really our first stage that we kind of begin this process with, that's the lone star. It occurs in the cytoplasm or cytosol. Okay? By the time glucose gets to this last stage, it's completely broken apart. We've produced the ATP we need, and we go about our business of doing those special things. So again, that obviously we need glucose to get into cells It'll be broken apart so that we can form ATP. Remember, we said to do several different things in our cells. For example, move muscles, get things across cell membranes by active transport, build molecules that our body needs. To do that, glucose has to go through 4 stages. You should recognize the stage names: glycolysis, preparatory steps, citric acid cycle, electron transport system, or chain. You should know where the stages occur to produce this ATP. Glycolysis occurs in the cytoplasm. The other 3 stages that you see here occur in the mitochondria, hence why the mitochondria is called the powerhouse of the cell. The rest of the stuff that I've indicated in my PowerPoint packet and that your book kind of goes through, you do not need to know. There's a lot of stuff here. If you take a higher level biology, if many of you are moving into Bio 144 eventually, then we're going to get really down to the nitty gritty. But right now, we're just overviewing it. If you just know the stage names, where things are located, and kind of the basic process of we got to get glucose into that cell, and to transform it into ATP, and reasons why, moving muscles and getting things across cell membranes, etc., you are fine. That's all you would need to know. That's why watching review sessions are good in terms of that. Now, the last thing I want to do is show you a couple of videos I have. These are videos from what are called Crash Course on YouTube, and they're 11 minutes long. I'd rather play the video now for you here than have you just give you the link and hope that you click on it, because it forces you to watch it in terms of that. You certainly can start and stop it when you need to, but this, I'll show you the transcript really quick, which you can look at. This is going to kind of go through some of the history behind the cell, but some of the different parts that we've talked about. Now, you might see some things we haven't talked about, like centrosomes, and briefly mentioned cilia and flagella, but here's the major organelles we've talked about. You're going to see Hank. He's the star of Crash Course. He's kind of quirky, but I like him. He is pretty good at kind of briefly explaining things, but yet putting it into perspective in terms of how it really works. So he's going to talk to you, review with organelles with you again, and then we'll do a little review with him on transport, and that'll be it for today. So kudos to Hank and his Crash Course. Here we go.
Building block, the animal cell. Excellent.
Now, thank you, Hank. There were some things in the video that we didn't talk exactly about in class, including protein synthesis in terms of how ribosomes are involved with that. You don't need to be worried about that. And then Hank here at the end of the video gives some interesting information about mitochondria and how evolutionary-wise we can trace our maternal DNA back quite a number of generations. More for you guys to be interested in than me asking you on the test. But I think you got a good kind of review of organelles. Now the next video that we're going to watch, and I'll show you again the transcript if you like to read things. You can see I'm not going to show you this whole video, but certainly if you wanted to watch it, because some of the stuff in regards to transport proteins here is not something that we need to look at in great detail. So I'm going to show you the first probably 6 minutes of the, maybe even the first 5 minutes of the video, so you can get an overview of the transport processes that we talked about. So here we go, Hank.
Different materials have different properties.
Okay, so we're going to stop Hank there. We got a little overview of some transport processes that we talked about in chapter 3. And again, if you want to watch any more, you certainly can. Sometimes seeing in video format does help. So this is the end of the review session. It probably went a little longer than an hour, but certainly the 2 videos that are here at the end, you can start and stop when you want, and you can start and stop me when you want. In terms of that, I hope it helps. I think it will, and we'll see you soon for test 1. Have a good night.